Welcome to my talk on genomic imprinting and making healthy embryos from stem cells. So, I don't know if all of you were at the talk from Max, who already talked about how to, about like some stuff related to strong germline engineering, and one thing that we want to really avoid if we do things in humans. Wait, that's not. Yeah. So one thing we really want to avoid is these kinds of imprinting disorders that we get because no matter if we reprogram, so if we want to make embryos from strong germline engineering methods, most of these methods involve culturing embryos culturing cells in vitro, and that means we need to take stem cells and turn them into embryos again. And the way this naturally happens in a human is that we have the egg cells, or the oocytes, and the sperm cells, and they get produced through a process that demethylates all of the DNA, and then they establish these imprinting marks that are different between the mother chromosome and the father chromosome. And if we want to imitate this in vitro, then we need to know how these imprinting marks look exactly, because otherwise we end up with disorders like the one that once you see here, which is something that we really don't want to see in humans. So, for example, in cloning, there's about 1% of dogs that get cloned end up with a disorder like this one, and others have muscle problems, and this is really something we want to avoid. So, yeah, for people in the audience who are not super familiar with epigenetics, basically one of the nucleotides, cytosine, there can be another chemical modification added to this one, and there's enzymes in cells at all times that both remove and add these chemical marks, and at different places there's basically an equilibrium where either everything is all methylated or it's all unmethylated. That's so every single one of these methylations is not super stable, but all of them together reinforce each other because the enzymes also read if things nearby are methylated, so that makes them more stable. So, for example, imprinting marks stay in some tissues, stay as long as the human is alive. So yeah, Max already brought this up yesterday, but in mice, it's easier to keep cells in a naive state in which it's easier to clone them. And this is called naive embryonic stem cells. And for the cells where this is harder, where the cells are still a little bit more specialized, this is called primed stem cells, or primed embryonic stem cells.
Can you quantify at all the high and low clonogenicity? Like what is like? Just like order of magnitude. You might not know that.
Yeah, I don't know if there's like any at all, or in humans it's not something that we really tested, but with, I'm not sure.
Okay.
But or high still means that, well, if you if you do SCNT in dogs, it works in like 1%, something like that.
What is low clonogenicity? Like, how often does cloning succeed for low clonogenicity?
Yeah, I'm not sure.
Okay.
Less than 1%.
Yeah. So yeah, here's another chart where you can see all of the marks on the DNA get basically erased where in the germline where you produce the eggs and the sperm, and then after fertilization, a lot of them get erased again, but not all of them. And then in somatic cells, the methylation goes up again. Except this is not here, but in the placenta, it stays a bit lower. It stays at like 50%. This is also one of the reasons a lot more imprinting marks get erased in the cells in the inner cellular mass because they get so methylated, but the ones in the placenta often stay around for longer, basically. So imprinting was discovered over 40 years ago now, where people did experiments where they took a zygote after fertilization, where you have the pronuclei from the mother and from the father, and then they used micromanipulation to switch around the pronuclei between different eggs. So you can switch out one from a father, and you can put in a new one from a different father and see if the embryo is still developing. And if you just take one out from a father and take in a different father, then it will still give you live offspring. But if you have two fathers or two mothers, then you get problems. And that's how people initially figured out that something is going wrong and something must be different between the chromosomes. So one tendency here is that when both are from the father, there's overgrowth, and when both are from the mother, there's too little growth. And the best explanation that we have for this is that there is competition between the mother and the father, because most species are not completely monogamous. So the mother is all the children that she has have her genetic material, but the father doesn't know if all the children that the mother is going to have will have their genetic material. So the father has less incentive to care about the future children than the mother. So the mother wants to keep more of the resources than the father. And the father wants to invest more in this current child. So in equilibrium, that leads to the father a lot of genes that would otherwise be expressed in both chromosomes. If the conflict is too big, some growth hormone is only expressed from the father's side and not from the mother's side. So in mice, then the early 2000s did a lot of experiments where they took cell lines from mice, where they only switched out some parts of the chromosome, such that some part of the chromosome is now from two fathers, and the rest is still normal. And then they grew mice from that and observed how the mice were different. And this is how they found all the different regions where we have imprints. So what you can see there is that there's these clusters where there's a lot of imprinted genes, but there's also some that are basically on all chromosomes, and it looks relatively different. That general pattern is the same, although it's all like different chromosomes, since it's all switched around in humans. Yeah. So in humans, we can't do the experiments where we just intentionally switch around the chromosomes, but sometimes this happens by accident. And in a few cases, this leads to very extreme disorders that we have names for, like Engelmann syndrome or Prader-Willi syndrome. But for a lot of chromosomes, these are more unknown, and they're not described that often. So basically for chromosome 15, 14, 11, 7, 6, these are well described, and the other ones are more unknown what's going on there. So there's another paper that looks at the data from the UK Biobank and from the 23andMe data, and they just look at, is there people where it seems like they have both chromosomes from their father look the same? So as a hint that this person has UPD means uniparental disomy. So when you have when both of your chromosome 7 are from your father, then you have paternal UPD. And basically through the UK Biobank and the 23andMe data, we know that this also happens on all the other chromosomes, and roughly at the same rate. Actually, chromosome 16 is the most common, but the symptoms are way less on chromosome 16, so most people who have UPD 16 just don't know about it. But it's not too bad to have this on one chromosome, but the problem would be that if we erase all the imprinting marks, we would erase it on all chromosomes, and then you have all of them at once, and that might be quite bad.
Excuse me, could you restate what UPD stands for?
Uniparental disomy. Yeah, so another thing with methylation is that there's a lot of there's lots of regions on the genome where there's all these regions from retrotransposons where the genome is actively trying to suppress the expression of retrotransposons, and those are also associated with methylation. In those cases, methylation is always suppressing the expression of genes, so people often associate methylation with suppressing expression, but with imprinting, there's essentially almost no correlation between whether the gene is expressed and which chromosome is methylated. It's actually the case that almost always the mother side is methylated and the father side is unmethylated. And this is one of the exceptions where the father side is methylated, and it's also the most well-studied example of imprinting, where we know that there's a protein CTCF that is binding better if the region is unmethylated, and they're changing how, I think there's another protein that binds to CTCF to curl the DNA around in a way that the enhancer is not reaching IGF2, and on the other side, the enhancer is able to reach IGF2, and then IGF2 is getting expressed from the father side. IGF2 is an example of a growth hormone. So this also shows that the growth hormones are always on the father side. This is how if you actually look at the raw data, how these actually look in practice, where it's a lot more messy. And this is actually not the region we just looked at, because so it's actually on the gene IGF2, where because the gene is getting expressed, when the gene is being transcribed, that also changes. So often when gene is being transcribed, there's some regions that get that get methylated. And you can see that when you look at the HG002 mat, that's a maternal chromosome, and the HG002 PAT is a paternal chromosome. We see the paternal one has a bit more methylation. So it's basically like this region. And also in sperm and oocytes, you see the same pattern, although in this case it doesn't say that much because oocytes are usually so this is also another problem. So one way you could figure out which side is methylated is you look, is the sperm methylated or is the oocyte methylated? But the problem is that oocytes are way less methylated than sperm. So you get a lot of false positives where you think that something is an imprint if it happens to be that in the somatic cells the methylation is about 50%, and then you think that there's a paternally methylated imprint, but actually it's just noise in the somatic cells, and then you see sperm are almost or everywhere very methylated and the oocytes aren't, and then you get a lot of false positives. The way to avoid this is to do phased reads instead. So this works really well when you have long read sequencing methods that can also things like nanopore, where you can both read the methylation, and because the reads are longer than the average distance between SNPs, you can figure out, okay, this read is from this chromosome and this read is from the other chromosome. And even better is if you have data from the parents, then you can even figure out if it belongs to the father or the mother chromosome. And this is basically what's happening with the HG02 maternal and paternal line there. Yeah. Yeah. So here's another tricky example. That's this IgDMR. It's one of the most important DMRs, and it also shows how. So we have very little oocyte data, and this is from this here on the oocyte thing is I think from like about 10 oocytes, and we don't have a single read from that region because the way that epigenetic reading is usually done is with bisulfite sequencing, where you add sodium bisulfite to your DNA, which turns all of the cytosine that's not methylated into uracil, and all the cytosine that's methylated doesn't get turned into uracil. And then you use this trick to distinguish methylated and unmethylated cytosine. But adding the sodium bisulfite also destroys a lot of the DNA, and then you get oocytes are already really expensive and have very little DNA, and then you also add this chemical that is destroying most of the DNA, and then even the reads that you're getting are so messed up that you can't properly align them. So in the end, you get very little data, which is one of the problems if you want to figure out where those imprints are. And that's this is where it's useful to have these long reads that you can do in adults or in embryos. The only problem there is that things might have changed. So for example, a lot of imprints that would be visible in the early embryo are being lost once you look at more specialized cells. So in this example, so the HG5 and HG2 are both from some type of white blood cell, I believe. And in one of the patients, the imprinting is basically lost, and in one of them you can still recognize it. It's also one of the smallest imprints. So this is just like, a bit more than 400 base pairs. Most imprinting regions are larger than this, more 1,000 base pairs and upwards.
So if it's so like in the second patient, or in the, I guess that's the first patient who doesn't. Oh wait, can you? Oh, in the second patient, like what's being sequenced there? It's a biopsy of like a whole bunch of cells. So does that mean the whole tissue like somehow lost the imprint?
So yeah, this is a bunch of different reads, and this is the average of how often it was either methylated or unmethylated. So
Does that mean like the whole tissue would have lost the imprinting?
yes.
Okay.
The cells are also, in this example, the cells are also cultured before they sequence them, so that might also make a difference. So you don't even know, is it the culturing or the or the replication that happened in vivo? Yeah. So basically, I think long reads are great, and we should do them in more tissues so that we figure out more of these imprints. But with the data we have so far, we can already make a rough guess how many of those different. So there's a difference between on the one hand, you have different genes that are being expressed from methylation, and you have these differentially methylated regions. And it's often the case that one gene that is imprinted, that is only being expressed from one side, there's more than one differentially methylated regions, and also a lot of non-coding, for one coding gene, there's probably a few non-coding RNAs that are involved in regulating the expression of that gene that are also being differentially expressed depending on which chromosome you look at. And yeah, in this study from Akbari et al., they find about 3. Yeah, it depends how you, wait. Yeah, I think they find about like 300 of them, and there's probably some. It's not entirely clear to me which of these are false positives and false negatives yet, because I haven't looked in all detail at all of these. But it seemed like if you compare the long reads to just looking at the percentages with when you look at oocytes and sperm, you get way less of this problem where you get all these false positives because the sperm are always methylated. Yeah, so this is basically great to have in addition to having more data from sperm and eggs. Yeah. Yeah, so this is another example where I mentioned earlier that the placenta is often gets less methylated. That means a lot more of imprints that would otherwise get erased because everything is getting methylated stay. And it's quite likely that they also have some function, because the placenta is where the placenta is in direct contact with the mother, and it's doing a lot of signaling to the mother how the embryo is doing, and it has it's directly extracting nutrients from the mother. So yeah, the placenta is especially important to get right with imprinting. So it's great that there's like already one preprint where people did long reads from 8 placentas in embryos and on fetuses? I'm not sure. And you can see like the upper example is a very classic imprint where the mother is 100% methylated and the father is 0% methylated. But the nice thing that you have with those long reads where you can phase things is that you can also find more weird patterns where it's harder to explain what's going on. Yeah, another thing is, so in principle, you can also do phasing with short reads. It's just working less well. But another problem there is that the this is a bit different, but like if you had the raw reads, you could also figure out. You could also better figure out in which of those two scenarios you are. But for oocytes specifically, it's hard to get. The biggest study on oocyte methylation is from 2014, and it's in a Japanese gene archive where I tried to, but because I'm not in academia and I don't have some ethics board, they don't give me access to the dataset. But thankfully, the study I mentioned earlier, the one from Akbari, they have the percentages where you just have the methylations and not the raw reads. They have that in their appendix. They include the data from the 2014 study, and you can't find it anywhere else. So if you look into that, that's useful. Yeah. So yeah, the phasing is also useful when you want to, as I mentioned earlier, whether the gene is expressed and whether the gene is methylated is almost completely uncorrelated. So it's, if you want to mechanistically understand what's actually going on, it's also good to look at the RNA sequencing data. And if you do, if in this case they also had data from the parents and could figure out whether the RNA was expressed more from the mother and from the father. I think there's a few others papers that also look at this, but I don't know if they also had data from the parents. And this is also great too. So I believe they mostly didn't find that many new imprints that we didn't know. And this seems to me like a good sign that we mostly have found most of these imprints, but there's a few regions where we might have missed one so far. Yeah. So thanks for listening, and I hope that looking more into this and especially finding ways how we can actually steer cells towards, like, to reprogram cells that they by default already have, like that we can make eggs that already have the imprinting all correct, would be really useful to get healthy babies and healthy cows. Yeah.
Questions? Ben? Yeah.
So I read a paper, they found like 9 different sites and just brute forced tied methylase enzyme to a guide RNA and had it methylate stuff around that binding site.
Mm-hmm.
It seemed to work.
Mm-hmm.
And they got healthy embryos.
Yes. That's another. So one of the advantages. So if you were making sperm, it might be easier to make sperm with correct imprinting because most of the sites are already unmethylated. There's the Akbari paper mentions a few more imprints that are paternally methylated that I before didn't know that they were paternally methylated. But there's probably less than 10 of them. So you could, if you were making sperm, explicitly target those to methylate them. If
Make sperm, we have plenty of that.
yeah, that's the problem. With eggs, you have like more like 200 of them. So doing all of them might be a bit harder.
A loop of genetic engineering where you have cell culture and you've got just the wrong imprinting pattern in the dish and you want to turn it into an embryo.
Mm-hmm.
It's I don't know how else you would do that.
Well, sperm and egg somehow do it. So there's presumably some transcription factors or something else that you could find. I'm not sure. Yeah.
So, correct me if I'm wrong, but freezing embryos causes their methylation patterns to change, shall we say. Have we noticed any difference between people who are born with IVF compared to people who are born naturally?
Yes, people with IVF tend to have more imprinting errors, but I just looked at this earlier. It's maybe 10 times more likely, but I'm not quite sure if it's also just because people who use IVF already are older and stuff like that. So I'm not sure.
Thank you.
Also, most of data suggesting that came from older studies where they used older IVF medium.
Yeah.
Also, you got to differentiate between like the freezing thing. If you're just doing simple IVF, they don't really need to freeze and thaw the embryos again, versus like long-term storage where you do have a freeze-thaw cycle.
Yeah.
But the freezing impacts it. I kind of doubt that, but the IVF process, just having the embryo sitting in a dish for a while, probably is more to blame than freeze-thaw cycle.
Yeah, I think there's, with regular IVF, you had other things that you're worrying about.
Did you, we talked a while ago about whether you could do bisulfite sequencing in a way that has less noise. Have you learned anything? What's your current?
Yeah, so there was one paper where I thought they were trying to amplify methylation, and I was thinking, oh, maybe that could kind of work. And the same group switched from that because, yeah, I was also not quite sure whether that would work, and they decided that it doesn't work well enough. But they did continue trying to find better ways to do bisulfite sequencing, and they now figured out a better, so they still do bisulfite sequencing, but they use a slightly different salt. So instead of sodium bisulfite, they use, I'm not quite sure which one they use, but they use a different salt, and it seems to, they get less, the DNA seems to get less damaged. So I don't quite remember the name of the company, but you can ask me later and I can look it up.
So it was better, but do you have any idea how much better or how good?
Let me think. I think it might be like really like you for the same amount of error, you basically destroy almost none of the DNA. So it's pretty good. But also, those claims came from the company, so I'm skeptical. That's like my main thing. Yeah.
More questions?
So you look at the Oxford nanopore data. I was on the assumption that they didn't need to do a conversion step where they could read the methyl cytosine natively through the pore. Is that true?
Wait, which paper?
Not a paper, but the Oxford nanopores methyl DNA sequencing, where they didn't need to do a conversion.
Oh, yeah. If you do long reads, then you don't need to do bisulfite. But the problem is that the input requirements are higher, so you can't do it. You can only do it if you have a lot of cells. So you can do it in an adult, but you can't do it when you have a single cell, because there's like, in an oocyte, there's like, I guess like 3 picograms of DNA, and you need more like micrograms of DNA to do long sequencing. So that's way too little, and you can't amplify it.
More questions?
I've got more of a comment than a question.
Okay.
If you go back to like the second slide, I know we were talking about clonogenicity stuff, so I can.
Oh, that was actually not an example of clones. Is it maybe that?
No, second slide, not the one right after the fucked up caps.
Wait. Oh yeah.
Comparing prime to naive cells. Go more. One more. Yeah.
Oh yeah. I stole that from an old textbook. there might be stuff that's out of date.
No, it's fine. So clonogenicity is the ability of a single cell sitting on a plate to form a colony. So 1 to 2 to 4 cells. So I was just going to mention, like, that's the important part of when I was describing the method of editing,
Okay.
is that you modify a bunch of cells and then you scatter them across the plate.
Okay.
And it's much easier if they're naive because they can just form their own colonies. Whereas with prime cells, they get scared because they're alone and then they either stop dividing or they die or gene expression changes. So that's
Thanks.
one of the reasons getting
Yeah.
stable naive cells is important for that stuff. But that was it. Thanks.
I have a question. So my question is, have you found any scientists who are, like, this is really important for basically all of germinal engineering. Like, if you want to like change the DNA in a cell and then make a healthy baby from it, we have to understand the epigenetics of reproduction. Have you found any scientists trying to like systematically understand this? I mean, obviously there's scientists who are like running the like collecting the data from sperm that and eggs that you've been looking at and so on. But have you find scientists who are like trying to catalog and understand directly
I don't think I've talked to anyone that you haven't like already like emailed or something like that. So yeah, or that's already here. So or like yeah, I guess some people here are trying to understand it, but I don't know if anyone here is trying to understand it very systematically.
All right. Any more questions? No? All right, let's thank Tassilo again.